네, 그러면 발표는 영어로 하는데 혹시 질의 물어보실 거 있으면 and I'm Jong Kim, and today I'm going to talk about my very interesting research, which is called the representational learning of the resting state functional MRI using VAE. And of course, I want to talk about its application in others and beyond the fetal-neonatal neuroimaging as well. So as you may know, actually, I will just really talk about this introduction of the MRI and functional MRI. I guess everybody knows, but MRI actually one of the modality can actually capture this inside of the brain, what's going on, non-invasively. That's why it became very popular. And among many other actual modalities, especially with MRI, one called functional MRI, it's basically we just measure this brain image over time. Then we actually can see that actually the signal in the specific area is changing, or we call the dynamics of the signals in the brain activity. So we actually measure this brain activity or board signal as a functional MRI. So this is actually one person's brain activity over time. So you can see the pattern itself, it looks like a quite noisy, but if you look at carefully and took some time, then you will eventually realize actually there are some of the distinguishable patterns, which call like a co-activation patterns. Especially we can see some of the different one network, visual network, and so on. So basically the goal of this my project or in our neuroscience is trying to just build a good or better technique to extract the features from the functional MRI. So before touching my method itself, I just want to give what is the most popular method in the field. It's called the independent component analysis or ICA. Basically, if we have a functional activity or the fMRI data, we actually can decompose the fMRI data into ICA map and the independent components. So what people found is actually the IC map, each map looks like a brain network. For example, actually, if you look at this bottom figure, the left panel is actually the IC map from the resting set activity, and the right one is actually during some specific task, their activation maps are showing. And what we can do is actually, even during the resting state, which does not involve any task, we actually can extract several different brain networks. For example, like a visual network, or I think maybe it's like a secondary visual network, default network, and so on. So this IC map is good enough to investigate several neuroscience questions. But going forward, actually, we actually know that the source of the fMRI data is actually not just based on this electrophysiological signal, which called the brain networks. On top of that, there are some of the like arousal levels and physiological signals not related to neuronal activity. For example, like a thermal noise, low frequency driven, cardiac rate, and so on. Plus, if you for the fetal and the newborns, there is also the developmental trajectory. And it's very natural that actually all of those sources are converged in non-linearly. Thus, we can think that actually resting set fMRI data is a highly non-linear. So basically, my research idea starts from here that we want to build a degenerative model that actually can understand this non-linearity of the functional MRI. So then why do we care about this variational autoencoder or degenerative models? Because so among other methods, I actually focus on the variational autoencoder. This is because actually the VAE actually can extract some of the linear features, for example, like a smiling and eyeglasses in the linear format, especially in the latent space. So this is how this is because possible because there is nonlinearity itself inside of these convolution layers, which called activation functions. So brain is nonlinear, so that's why the nonlinearity implemented in the deep learning algorithm can be a key to understand how the brain is activating. So the learning for learning representation of the adult functional MRI data. And then we characterize the representation of the fetal newborn fMRI data using the VAE trained on adult fMRI data. And lastly, this is more like a neuroscience question that we want to investigate what is the individual signature in the newborns using the VA models. So then, I'm sorry, okay, I'm sorry. Okay, so then what's the difference in the fMRI data and natural image, which was widely used in the VAE? So actually, she's my daughter, Luna, and for most of the cases, the natural image is presented in the 2D space for most of the VA model. However, the functional MRI is itself as a 3D. That's why this one layer or one more dimension in the input space actually introduce a lot of complexity in the models. Plus, on top of this one, we actually know that not just different from natural image. Actually, for brain, there is a circus and gyrus, and actually gyrus is highly convolved across this outside of the cortex, which actually it's very different. Is that so even though within the very neighboring pixels or voxels, they can have a very different properties. For example, white matters and the gray matters, or even though it's a gray matters, if they're in the different layers across the circuits, it actually can have a different properties. Of course, actually, VA model itself can have a mechanism to learn those kind of structural differences for example, in the gray matters. But however, then actually introducing that one to the model actually provides more complexity in the model. That's why we have to make the VA models deeper and the more, which means the more parameters that for learning, which means that we need more data sets. So when we are during this designing the model, what we wanted to do is that we call the geometry reformatting. Basically, we just did not want to take this 3D volumetric space as an input. Instead, we actually can present them into the cortical space. And again, the cortical space can be presented as a spherical model. And spherical models can be presented as azimuths and elevations. Then if we present them azimuths and elevation, we can actually plot them as a 2D image. So what we actually found that is once we actually convert or reformat this volumetric space into this 2D space, actually the training time of this significantly reduced from, for example, over 100 hours of this training time for 3D volumetric functional MRI versus only 5 hours for 2D cortical image. So we actually was very excited with this geometry reformatting, and we really hope that actually this is more like a ground for the deep learning applications for board signals or functional MRI data. Anyway, so after we built this geometry reformatting, we also designed some of the architecture of the variational autoencoder. And among other types of this VA model, we used the Beta VAE. And so Beta VAE basically consists of the 3 parts: the encoder and the latent variables and the decoder. And this is actually the governing loss function for the VA model. Basically, it has 2 parts. The one is the model for model performance. It's just sum of the squared errors plus the beta multiplied by this model constraint, which is reflected by the KL divergence. Basically, we had to adjust these beta values to make this optimal place for model performance versus the model constraint. And again, this encoder had 5 layers of convolution layers, and the decoder had 5 the transpose convolution layers as well. And we actually had 256 of the latent variables. we are actually very interested. And then once we built this VA model, we actually tested and trained using the HCP dataset. Probably you guys have heard about it. So basically, this HCP dataset has a lot of this the board signal or functional MRI data from more like 1,200 subjects. So among 1,200 subjects, we used the 100 subject data, which is each subject has 1,200 time point of the fMRI data. And then we use the validation data set for 50 subjects. So we actually determined the optimal hyperparameter of the VA models. And then finally, after we well trained the model, we actually first we wanted to look at whether actually the VA model can compress the activity of the resting state fMRI data. So this, for example, this is original fMRI data, and after through the VA model, we actually can see the pattern is a little bit more like a smoothed version of the original image, but still we can see the patterns quite looks similar. And surprisingly, this is actually the black one is from the original fMRI data in this green area, and actually the red one is a reconstructed fMRI activity, and you can see it actually follows pretty well, suggesting that actually it still can get this temporal information of the fMRI data. And then we were actually curious then whether, since this VA model itself is generative model, what is the meaning of this generation of just from the random signal? So basically, we just get the total random input, which is just one standard deviation plus the zero mean, and then just as a reference variable, and we put them in as a decoder, and just synthesize the no meaning random fMRI data. But what we observed is actually once we build this synthesized fMRI data, we just can see that actually the seed-based correlation patterns, putting this V1 as a seed region, it looks like a visual network. And for dorsal rotation network and deep mode network, which are actually very highly resembling the actually measure the cortical activity patterns. So we are very excited that actually I think a VA model can synthesize more like a structural functional MRI data in decoder. And next time we want to look at whether how we actually can cluster the latent space. So for this time, we get this input from the 500 subjects and then put them all of the encoders. And then in the latent space, we apply this k-mean clustering that we cluster to each clusters. And then we took this centroid of the clusters. And again, using the decoder, we look at how the pattern of the centroid of each cluster. And this is actually the reconstruct images of this centroid. And you can see their patterns looks like a brain network. For example, one is like a default bond network, and the 21 is more like a motor network, and the visual network here, and so on. And I will not go details about this each network, but I just want to give one example of network that I think we were very highly excited. The number 2 is actually consists of the very focal regions. For example, is like a MFG, premotor area, supplemental area, the primary sensory area, and also cingulatory motor area, which is very well differentiated here. And plus, actually, the primary motor area was actually separately presented as a different clusters. So, and then so far we only look at these group level features, but instead we also want to look at whether actually the VA model can preserve this individual information or individual variations. So we actually took these 20 subjects of the fMRI data and then again put them in encoder. And then we just plot them in the 2D space using the TSNA method, or called the T-distributed stochastic narrow embedding. But what we actually observed is actually their patterns are well differentiated from individuals. You can see all of them. However, it was not the case for the ICA. You can see all their messed up here. So we want to look at whether actually this information, what's called individual variation, presented in the latent representation, can be used for individual fingerprinting. So basically, the individual fingerprinting, how we actually do is that we measure the functional connectivity patterns, which is just a correlation between the different latent variables or ICA maps. And then we actually took that. It's more like a pattern of the functional connectivity is used as a fingerprinting of somebody. So for every 500 subjects, we actually have a one first session, and after a few weeks later, they actually took the fMRI data again, which is called the session 2. And then we use the session 1 of the data as their fingerprinting and try to identify somebody only based on their patterns. And what we actually observed is that the accuracy of this identification is almost like 97% among 500 subjects. And then we just want to compare it between the cortical space and the latent space. Cortical space actually either is ICA. So you can see if we increase the number of the subjects for identification over and over, then their accuracy is going down very rapidly. However, it was not the case for our latent space. We actually see the accuracy is always almost a 1, it's all 100%. So that means that actually the latent space can be used for real fingerprinting tasks. Plus, they not only based on this number of the subjects, even with a very shorter data point, for example, like only with 3 minutes, we were able to identify subjects very highly, for example, almost like 90% of accuracy. With only in the latent space versus, for example, in the cortical space, you only reach the 30% of the accuracy. So this is all based on these others. And the take-home message on this part is that first, the latent variables defined by this VAE represent fMRI data better than linear compression methods. Second, the latent representations can be clustered by brain states. Then lastly, I think it's interesting is that the representational geometry, which is like a functional connectivity patterns of the latent variables, can be used as a reliable neural fingerprinting of the individuals or potentially a biomarkers of brain diseases. Actually, before going forward, if you guys have any questions, please let me know.
네, 좋은 강의 감사드립니다.
아니, 저 더 있는데.
네네.
그럼 질문 없으면 더 계속 진행하겠습니다. So, and then now we built this the VA model, and now actually I moved to this Children's National Hospital. I still wanted to pursue how we can use the VA model for fetal newborn functional MRI. So basically, research core was it's more like exploratory study that we wanted to use the VA model which was built based on the adult fMRI data, applying to this fetal and the newborn fMRI data. So actually, we used the 2 sets of the fMRI data from our own institute. I will call the DBI data set, consisting of the 110 fetuses and 139 newborns. And the data length was about 5 minutes. And we also used the public data set from the DHCP. Basically, it consists of over 400 newborns, and all of the age range was like between the 24 weeks to 42 weeks. And it was about much longer time point, which is like about 15 minutes. And again, the first question is actually whether the VA model still can be useful for representing newborn resting sensitive functional MRI data. And this is actually the original image and how we actually reconstruct the image from the original, either using the VA model or different IC methods with a different number of the compounds, 50, 100, 200, and 300. again, the latent number of the latent variables or components of the VA model is 256. So I guess the performance is supposed to be between the 200 and the 300. But surprisingly, actually, the VA model showed the best reconstruction performance, suggesting that actually still the VA model, which was trained based on this adult fMRI data, can be still useful for understanding the newborn resting fMRI data. And this was not only for the case for the DHCP, but even for the DBI data sets. And then now although we have more data set to validate the usefulness of the VA model, but this is more like, I think it was very interesting result I want to share. So basically, we want to use the VA model to get this network mappings of the newborn or the fetuses. So basically, for fMRI data, we also encoded, generated as a latent variable using the encoder, and we apply this temporal ICA and get this IC map, and then again we look at the pattern of the network using the decoder. And very interestingly, actually, this is some of the examples of this network generated from DHCP full-term newborn. And the 8 one, the number 8 looks like a visual network, but it was a contralateral visual network. And a bilateral auditory network. And again, maybe it looks like a kind of different mode network, but again, it's a contralateral. And surprisingly, actually, those kind of brain networks we observed was actually reproduced in the full-term newborn, but from the different or the DDI. And what we observed is that this pattern similarity was actually highest between this full-term and the full-term across the DHCP and the DBI compared to other data sets, for example, like even within the DHCP data set, but if you use a comparison between the preterm versus full-term, actually their pattern similarity was lower, which means that actually the VA model not just get these patterns from these pre-processed steps or difference in the pre-processed steps. They actually look at this neuronal activity or neuronal patterns from this fMRI data and look at what is the similarity in their patterns. And this was actually surprisingly, it was not the case. So if you look at this inter-data set pattern similarity, which is the black, versus the intra-data set pattern similarity, but across the different age groups, for preterm versus full-term, or fetus versus full-term, actually only VA models show the higher pattern similarity. But for example, IC300, IC200, IC50, they show the more similar across within the data set, but across different age groups compared to within the same group, same age group, but across different data, suggesting that what we can consider the VA model can be used as a more like a general tool to get this brain networks across the different data sets, especially when their different data sets are pre-processed using different pipelines. So the take-home message here was that actually the VA model was generalizable across the age groups and data sets. And the learned representation using the VA model is actually explainable as a cortical network and the dynamics. So this is the final part of my study that I want to show that investigating individual signature in newborns using the VA model. So as I mentioned, in adults, everybody is unique. Basically, you can identify somebody based on your brain wiring patterns. The accuracy was like 98%. However, in previous 2022 study showed that in newborns, actually subject identification task was very low, which is about just 10% among the 31 subjects. And they conclude that actually in newborns, their functional network patterns are not unique to individuals. They're just more like showing this group level analysis. However, actually, in behavior studies, they already show that even newborns, from the early born, they already show their unique behavioral pattern. So we were thinking maybe this is because the how they define these features or functional connectivity using the ICA or this cortical parcellation was the problem. So this is the reason why we wanted to argue is that unlike adult age growth, actually newborn brain grows very rapidly. And as I mentioned before, so basically how we do this task is that you get this one task, one session of the fMRI data, and 2 or 3 weeks later, you acquire the fMRI data again. And then you think that actually their brain patterns are always the same. Probably that's the case for most of the age groups, but not for newborns, because for newborns, just 3 weeks, their brain is growing very rapidly. So basically, there is a very strong age-related dissimilarity effect between groups. So we thought actually the reason why the previous study failed to identify somebody based on the brain wiring pattern is because they did not consider this age-related dissimilarity. So again, we used the DHCP data set, and we used the 361 newborns with a single session, plus the 48 newborns with the repeated sessions for a subject identification test. And what we observed is actually, so we actually try to predict the age with the functional connective patterns, either using the VA model or cortical atlases for like a VDG, RSN7, RS17, or different GI group level ICA with a different number of the IC compound, and the group level analysis from these young adults with a different number of the compounds. And what we observed actually for age prediction, that even with the ICA, their performance is comparable. However, for individual identification tests, actually only VA model showed very high accuracy, about 47%, compared to other methods. For example, GICA, the best accuracy when you use the ICA is about 30%. And then we further question, as mentioned, actually there is a effect from the aging. So we wanted to separate them, whether we were actually curious whether we actually can separate of the functional connectivity that is relevant to aging or age independent functional connectivity. So basically, so since we have this single session data set, we just try to predict some of the functional connectivity, and based on that, we identify functional connectivity patterns. And so and then based on the age prediction performance, we divided them into the 10 groups, which called the connections relevant to aging. The left is the most relevant to aging versus the least relevant to aging. So of course, the connection relevant to aging show the highest age prediction performance. However, you can see the subject identification accuracy was lower or lowest. And then if we go to connections with less relevant to aging going further, what we observed actually subject identification accuracy is going higher, but at the same time, age prediction goes lower. And we can see actually there is a very clear crossover between the age prediction performance and the subject identification accuracy. Again, it was not the case for cortical parcellation and the ICA. Suggesting that actually the VA model somehow actually separated those kind of age-relevant components and this individual uniqueness, the features, and separate in the latent space. But it was not the case for the cortical parcellation or GICA. So finally, the summary of my finding was that the functional connective fingerprint appeared to be actually established during the newborn period. However, unlike other age groups, FC change from the scan interval then also in the VA-derived nonlinear space, age-related effect was effectively isolated, but it was not the case in linear space. So thus the VA model can be useful tool for newborn fMRI studies. So this is all of the one that actually I worked in, but on the way, there are several ongoing VA projects. One is the one I'm working with the Cindy, which is a PhD candidate from the WashU. Basically, instead of we put this fMRI activity patterns, we just want to give the functional connectivity profile as an input of the VA model. And we observed when we give this functional connectivity as an input of the VA model, and the network patterns are very well separated and segregated into different modules. For example, like visual network and default network and those things, especially across the different data sets. For example, Human Quantum Project or Midnight Scam Club and the Baby Quantum Project. And we actually submit this work into Imaging Neuroscience, and it's just under the second revision. And hopefully this paper will be accepted very soon. And the other line of the work is that we want to also predict this fMRI dynamics using the VAE and plus the BERT. And for this project, I'm working with Owen. He is a PhD candidate from University of Michigan. So still it's a very preliminary. We found a very interesting point is that when we introduced the BERT models, after this VA model, actually BERT model was very highly predict the activity of this latent variable, suggesting there is a potential that we can use the BERT model to either predict the fMRI dynamics, or we actually can use this one to reconcile of this when some fMRI activity pattern was contaminated by motion levels or head motions. So this is all about my research, especially using the VA models. But I just want to very briefly touch on what is my other research interests. So again, I'm the machine learning and the deep learning person. So I'm actively developing advanced pre-processing pipeline for fetal newborn fMRI data. For example, the volume sensing method for fetal and the newborn fMRI data. And also I'm using some of the machine learning applications for fMRI data for like prediction of the GA using the gestational age, using the functional fMRI data. We look at cerebral volume and the functional connectivity in newborns, and so on. And finally, I'm also interested in the neuromodulation. So during my master degree pursuing the master in the Hanyang University, I was focused on the TCS, and then now I'm working as I'm working as a PI in our lab. So I'm working with some of this research faculty in our lab. We submitted R21, it's for about identifying these multimodal factors that determine the individual response to TCS. And surprisingly, this R21 grant was accepted very recently, even after considering all of the challenges, the NIH and so on. And plus, our interest, the proposal was also accepted in the RCP, which is Rainer Cerebellum Project. It was for $5 million for 5 years. And I'm enrolled as an AI ML key person. And finally, as a PI, I'm also interested to laying the groundwork for safe and effective T-cell system in infant. As you may know, actually T-cells never introduced the infant, and the main challenge is because we don't know whether it's safe or not. And so A1 is about this establish dark electrical conductivity values for infant scar tissues. Basically, I'm collaborating with a neurosurgeon in our hospital that we are trying to collect these infant scars during their surgeries, and then we build by building this measurement experiment. We want to measure actually what is the conductivity values of the infant scars. And based on that, we want to build this individualized 3D model of the infant health so that we can simulate the T-cell current flow. And again, the last one is during my lab, during my PhD, which is called Libby Lab, and he's a Dr. Zhongmin Liu. And right now it's our DBI lab. We are actually very big. We have about over 60 people in our lab. And this is our funding. Especially, I love to say thank you for NICHD during my postdoc T32 training grant and or others. Thank you. And just let me know if you have a question. Thank you.
네, 강의 감사드립니다. 그, 뭐, 질문이나 코멘트 있으시면 부탁드리겠습니다. 음. 우선은 제가 그냥 한 가지 질문 드리고 싶은데요. 이거는 이제 교수님 하시는 연구 그 내용 컨텐츠와는 직접 관련이 있는 건 아닌데 이제
네.
주되게 이제 연구 관심 분야가 말씀하시는 게 베리에이션의 극복 차원에서 이제 개인차에 대한 이제 그리고 아기들이 이제 빠르게 브레인이 발달을 하기 때문에 형태도 바뀌고 기능적으로도 굉장히 빠른 시기에 바뀌기 때문에 이런 베리에이션의 극복 차원에서의 이제 포커스를 두시고 연구를 주로 하신 것 같은데 한 가지는 최근에 이제 MRI 장비 간에도 차이가 있어서 빅데이터 분석을 위해서는 이제 대규모 연구 진행할 때 허들이 되어서 하모니제이션이 굉장히 중요한 이제 요건으로 고려를 해야 되는데 이 교수님께서 하시는 이 방법을 하모니제이션 개념의 파이프라인으로도 적용이 좀 가능하지 않을까 싶은 생각이 잠깐 들었는데요. 혹시 어떻게 생각하시는지 여쭤보고 싶습니다.
좋은 질문 감사드립니다. 사실
네,
저희 지금 저희 DBI에서 하고 있는 그 멀티센터 프로젝트가 있거든요. 제
예,
오보라고 되게 재밌는 프로젝트라서 이게 잠시 소개드리고 싶은데 오보라고 이제 opioid exposed newborn study인데, 그러니까
예,
그 임신 상태일 때 특히 미국이니까 그 오피오이드에 중독된 맘들이 많으니까, 그래서 그 태아 있을 때 그 오피오이드 익스포즈가 됐을 때 그러면 뉴본이 나왔을 때 그때 브레인이 어떻게 차이 나는지를 보는 연구거든요. 그리고 이 연구가 이제 센터가 네개가 있는데 제가 DBI 하나 있고, 그리고 보스턴 칠드런 하스피럴 있고, 다른 두 군데도 신시내티 칠드런 하스피럴인가 이렇게 여러 군데가 있는데, 거기서 저희가 지금 그래서 제일 고민하고 있는 게 그 하모네이션 그 네 개 센터에서 하는 거거든요. 그래서 일단 저희가 느낀 거는 일단 그 에큐지션 파라미터들을 일단 다 동일하니까 그래도 어느 정도 그 인터센터 베리에이션이 있더라고요. 그래서 그걸 하려고 사실 딥러닝 베이스는 아니고 그, 그냥 이렇게 컨벤셔널 메소드에서도 저희가 이런 식으로 해서 그런 걸 차이를 보는 연구를 진행하고 있습니다.
네, 네, 감사합니다. 예, 저희 채팅창에서 이제 황선욱 부학장님께서 이제 질문을 주셨는데요. 우선은 제가 읽어드리면 이제 몸담고 계신 국립연구소에 대해서도 조금 더 자세히 부탁드려도 될까요? 그래서 이를테면 박사님 같은 전문 인력이 많이 연구 활동을 하고 계신가요? 그리고 소아 연구를 하실 때 의료기관과 연계하시거나 임상의사가 참여해서 IRB 절차를 받으실 필요가 있나요? 그리고 그러한 허가 절차는 보통 얼마나 소요되나요? 뭐 이렇게.
아니, 질문 하나씩
네,
말씀드리겠습니다.
네, 네.
그, 미국에서는 예를 들어서 제가 알기로 한 칠드런 하스퍼들이 리서치 베이스 칠드런 하스퍼들이 한 여섯 군데, 일곱 군데 있는 걸로 알고 있어요. 그 중에 저희가 하나고, 거기서는 그 리서처들이 꽤 많은 거죠. 특히 저희 같은 DBI 연구에서는 저희가 한 60명 되거든요. 그 중에, 근데 사실, 확실히 그건 다릅니다. 그러니까 60명 중에서 이제 리서처들은 이제 20명 정도 되고, 이제 20명은 클리니션들, 그리고 20명이 이제 리서치 클리니션 이제 코어디네이터들이거든요. 왜냐면 사실 이게 리크루팅이 정말 쉽지 않은 것 같아요. 특히 왜냐면 그 이제 성인을 대하는 게 아니고 이제 그런 이제 이런 이제 엄마들이 와야 되니까 그런 걸 이제 리크루팅 하는 게 쉽지 않아서 그래서 그게 제일 힘든데, 그래서 그래도 연구실에 연구하는 사람들은 꽤 많은 것 같아요. 예를 들어 저희 DBI만 해도 그 패컬티가 한 15명, 리서치 패컬티만 15명 있고, 그리고 저희 병원에 센터가 5개 있거든요. 그 센터 하나는 뭐 저희처럼 이제 뉴로이미징 하고 뭐 클리니컬 트랜슬레이션 하고 이제 그리고 서지컬 이노베이션 있고 뭐 이런 식으로 있는데 연구는 다양하게 하고 있습니다. 그리고 IRB는 어 그래서 사실 저희 연구실에도 그렇고 그 IRB를 따로 이렇게 스페셜하게 해주는 분들이 있거든요. 그러니까 당연히 리서치 아이디어는 저희가 들고 가죠. 대신에 이제 그런 페이퍼 워크나 이런 것들을 이렇게 옆에서 도와주고 이제 프로세싱도 저희가 이렇게 빨리빨리 진행할 수 있게 해주는데 그래서 보통 IRB 그 스크래치에서 이제 처음으로 받는 신규로 받으면 한 6, 7개월 정도 걸리는 것 같아요. 저 같은 경우에도 그 제가 지금 이번에 한 그 인펀트 TCS 그 스컬 받아서 그 일렉트릭 컨덕티 하는 것도 한 최종 한 6, 7개월 정도 걸렸습니다.
음,
그리고 그리고 사실 그 저희가 DBI 랩이 그 센터에 이렇게 엄브렐라 밑에 있거든요. 그리고 그 다른 랩 중에 하나가 이제 PPI라고 프리네탈 인스티튜트가 정확히 모르겠는데 그 PPI 연구실에서는 이제 이지도 엄청 그 이지를 사실 제일 많이 하거든요. 그래서 거기는 이제 센터 디렉터가 아니 랩 디렉터가 이제 그 닥터 메디컬 닥터라서 실제로 그 이제 그 니큐나 ICU에서 이제 실제로 EEG를 많이 찍으면 그 EEG들이 아예 자동으로 그 연구실 데이터베이스에 들어와서 그 프리프로세싱이 되고 그럼 이제 프리프로세싱 돼서 그 이제 그걸 이제 어널리스트 한 다음에 그걸 아예 그 리얼타임으로 이제 피드백을 주는 그런 시스템도 만들고 있어서 굉장히 액티브하게 하고 있는 걸로 알고 있습니다. 그리고 DB는 Developing Human Quantum Project, DHCP 데이터 같은 경우에는 사실 해외에서 가능한지는 저도 잘 모르겠습니다. 그, 왜냐면 특히 미국에서도 그런 해외 쪽에서 그 연구하고 하는 걸 그 되게 요새 다시 리이벨루션 하고 있거든요. 그래서 DHCP 같은 경우에는 이제 그 데이터를 받으려면 그 리서치 프로포저를 제출하고 그 DBI 그 크레덴셜을 받아야 되는데 아이디를 받아야 되는데 그게 외국 인스티튜트에서도 나온지는 잘 모르겠습니다. 그리고 사실 그리고 제가 이제 고백해야 될 것 같은데 그 저희는 이제 병원에 있으니까 리서처들은 그 MRI 애퀴지션에 참가할 수 없습니다. 이게 모든 그러니까 그 MRI 애퀴지션을 하는 왜냐면 특히 저희는 이제 신생하거나 태아이 경우이니까 이게 엄청 클리니컬하게 이게 되게 센서티브해서 아예 그 애퀴지션 하는 어, 프로페셔널들이 있어서 그분들이 이렇게 하고 대신에 이제 저희는 이제 프로세싱이나 뭐 저희가 뭐 예를 들어서 그 애퀴지션을 이제 파라미터 아니면은 뭐 그 MRI를 이렇게 새로 뭐 하고 싶다. 그러면은 이제 같이 해서 저희가 참관을 할 수 있지만 실제로 저희가 뭐 이게 그 실행하거나 아니면 찍을 수는 없습니다. 그래서 저도 그 박사 과정 할 때는 항상 FMRI나 펑셔널 MRI 찍고 EEG도 찍고 했는데 저희 지금 연구실에 와서는 참관만 해봤고 사실 실제로 그 MRI 머신을 해본 적 없어서 그건 이 질문은 조금 답하기 힘들 것 같습니다.
네, 감사합니다. 또 다른 질문 있으신 분 있으실까요?
제가 한 가지 하고 싶은데요, 김정 박사님. 그러니까 제 초반부 첫 번째 파트에서 3차원으로 만들어진 fMRI 볼륨 데이터를 쫙
네,
펴가지고 평면으로 만드신다고 했잖아요. 그래서 데이터 프로세싱 속도를 현격하게 이제 줄일 수 있다.
네,
그러면 아마도 그 2D로 다시 리컨스트럭션 한, 리포메이션 한 데이터의 픽셀당 원래 볼륨이 갖고 있던 XYZ 정보가 다 인코딩이 어딘가에 돼 있을 것 같은데,
네, 맞습니다.
그렇게 프로세싱 3차원으로 리컨스트럭션 된 거를 톡 찢어가지고 2D로 펼쳐서 프로세싱하는 속도랑 이 FMRI 만들 때 어쨌든 스택으로 슬라이스로 다 찍어가지고 합치잖아요. 그
네, 그렇죠.
2D 사진을 여러 개 스택으로 프로세싱 하는 거랑 뭐 데이터 처리 속도나 효율 아니면은 그 커넥텀을 분석하는 면에서 어디가 더, 왜 장점이 더 있는지 좀만 더 설명해 주실 수 있어요?
그럼요. 잠시만요. 사실, 혹시 보이시나요?
네,
네. 그, 제가 사실 장점만 말했는데, 예를 들어서 트레이닝 시간이 엄청 빠르고 한데, 사실, 길게 보면 3D 볼륨 스페이스에서 하는 게 제일 맞다고 생각합니다. 왜냐면, 일단, 이제 콜티컬 스페이스 넘기면서 사실 제가 말 안 했지만, 딥 브레인 리전, 뭐, 세레블럼이나 이런 것들은 다 저희가 이 2D 이미지에 없어지거든요. 특히 그 신생아나 뉴본 같은 피터리나 뉴본 같은 경우에는 세레벨럼이 제일 먼저 발달하는 걸로 알려져 있거든요. 그래서 그런 것 때문에 조금 리미테이션이 있다는 거 확실히 말씀드려야 될 것 같고 그래도 그 한 가지 제일 큰 장점은 뭐냐면 예를 들어서 이게 사실 3D 볼륨을 이제 2D나 뭐 1D로 만들려는 방법이 되게 많았고 예를 들어서 이렇게 볼륨메틱이 3D인데 그럼 예를 들어서 이제 그레이 메탈 볼륨만 이렇게 복살만 뽑은 다음에 그걸 그냥 이제 1D로 만들 수 있죠. 그렇게 하는 경우가 굉장히 많은데 근데 저희가 생각하기에는 그 방법은 사실 서버 옵티멀한 방법이라고 생각하거든요. 왜냐하면 이제 딥러닝 같은 거 해보시면 아시겠지만 이제 딥러닝에서 이제 리세트 윈도우가 있고 그게 복살 사이에 이제 네이버링을 커넥션을 봐야 되는데 이제 1D로 만들 경우에는 이게 바로 옆에 있는 복셀이 이게 실제 이제 완전히 다른 곳에 있는 거죠. 이게 사실 런 컴플렉시티를 다 당연히 모델이 만들 수 있지만 그렇게 하면 제가 생각하기에 좀 비효율적인 방법이라고 생각하고 그래서 이런 식으로 2D로 하면은 이게 예를 들어서 이렇게 보시면 아시겠지만 콜티컬 스페이스에서 붙어 있던 픽셀들이 아니면 버텍스 같은 경우에는 2D로 바꿔도 그 옆에 붙어 있잖아요. 이게 대신 그래서 이제 그 바이어스가 덜 생기는 방법이라고 생각합니다. 뭐 그래서 조금 더 말씀드리면 사실 이 방법보다 더 제일 좋은 방법은 제가 생각에는 이제 저희 같은 경우에는 이런 그냥 아, 딥러닝 베이스를 썼는데 이거 방법 말고 그래프 베이스 뉴럴 네트워크라고 있거든요. 아직도 그리고 그거는 이제 컨버전 예를 들어서 저희가 쓰는 이제 2D 같은 경우에는 컨버전 레이어가 이제 네모나게 컨버전 리셉팅 윈도우를 형성하는데 이제 그래프 베이스 뉴럴 네트워크 같은 경우에는 이제 그래프로 만든 다음에 그걸 이제 어떻게 하는지를 하는데 근데 아직 그래프 베이스 뉴럴 네트워크 같은 경우에는 컨버전 레이어를 정확히 어떻게 해야 되는지 디파인 하는 게 이게 쉽지 않아서 그래서 저 같은 경우는 이 방법을 쓰고 있는데 어, 그래서 저희가 생각하고 있는 여러 가지 방법들이 있거든요. 예를 들어서 지금 제 같은 경우에는 이제 2D로 만들어서 콜텍스를 이제 2D로 만들었고 그러면 콜텍스 대신에 예를 들어서 콜텍스를 한 2D로 만들고 그리고 그 서브콜티컬 리전들을 다른 하나의 더 슬라이스 픽처를 만드는 거예요. 그러면은 이제 콜티컬 스페이스 하나 있고 사실 레프트 헤미스피어, 라이트 헤미스피어 그리고 딥그레이 리전스. 그럼 세 장이 이제 하나의 인풋으로 들어가서 그럼 세 장 사이에 이제 피처들을 볼 수도 있고 그러면 이제 제가 생각하는 단점은 뭐냐면 그러면 이제 마지막으로 볼 수 있는 게 그 세레브럴 커넥션 예를 들어서 어떻게 그 코르티컬 리전이랑 그 딥브레인 리전들이 인터랙션을 하는지를 그냥 리니어하게 보지 않고 이제 라니니어한 인터랙션들을 레이턴 스페이스에서 확인할 수 있는 방법이라고 생각합니다. 그래서 좋은 질문 감사드리고 제가 제가 이 BA 모델을 이제 박사 때부터 해서 이제 한 6, 7년 하고 있는데 그래서 정말 많은 피드백도 받고 어떤 방법을 하고 했으면 좋겠는지 이런 것도 하는데 이게 시간 내기가 쉽지 않아요. 그래서 정말 극치 뭐 같이 연구하고 싶은 분 있으시고 뭐 아니면 VA 모델 어떻게 사용하시고 싶은지 있으시면 진짜 저한테 연락 주시면 제가 이제 성심터니컬 해서 알려드리겠습니다. 그래서 제가 지금 지도하고 있는 그 박사과정 두 명도 그 제가 제 한 명은 제 VA 논문 읽고 그 연락 와서 같이 그 박사 2년 거의 2년 반 동안 같이 연구 진행해서 이제 이번에 졸업하고 그 신디 같은 경우에는 그 오행 같은 경우에도 그 VA 모델 같이 하면서 이렇게 질문 이렇게 주고받다가 같이 이제 저 같이 지도하는 걸로 했거든요. 네, 그래서 그렇습니다.
네, 감사합니다.
네, 또 다른 질문 없으실까요? 네, 그러면 일단은 오늘 저 세미나는 이것으로 마치고요. 오늘 그래도 귀하신 시간 시차도 잘 안 맞는데 저 시간 내주셔서 네, 강의해주신 김정은 박사님께 감사드립니다. 그 말씀 주신 것처럼 좀 저희 고려대학교랑 같이 공동 연구도 앞으로 진행을 했으면 좋겠고 저희가 지금 뇌영상 센터가 새로 이제 좀 정릉에다가 생길 예정이어서요. 그래서 더 많이 관심도 있고 저희 학장님도 지금 참석해 주셔서 이제 관심 갖고 봐주신 것 같습니다. 그래서 저희가 뭐 요청
그렇습니다.
드릴 사항들이 있으면 공동 연구나 이제 테크니컬하게 좀 도움 요청 드릴 게 있으면 좀 이제 요청 드리도록.
네, 감사합니다.
네. 네. 오늘 좋은 강의 감사드립니다. 네.
네, 시간 내주셔서 감사합니다.
네.
네, 고맙습니다.
네. 감사합니다. 네. 이것을
네, 좋은
마치겠습니다.
아침 보내세요.
네. 네.
네,
감사합니다.
감사합니다.
네.